some of your questions. Thank you very much.
So thank you, Priya, for the kind introduction, and good afternoon, everyone. I am Veronica Venafra, and today I'll be talking about SignalingProfiler, which is a system biology tool to interpret multi-omics data, building signaling networks that are connected to functional cellular traits. So a little bit of introduction on system biology, which is a discipline that helps us tackle complex biological questions by aiming at reconstructing a holistic picture of biological systems using computational models. So in these computational frameworks, we can think of healthy or drug-sensitive context as the expected output, while we can think of disease or drug-resistant context as the altered output. In this computational model, we reconstruct the molecular mechanisms leading to the expected and the altered output, and we can compare the topology of these models to identify some topological differences that may represent some points of intervention that can help us to restore the normal function of cells or restore drug sensitivity. To build this network, we can exploit multi-omics data, which will provide an unbiased picture of the system. But we all know that omics data are noisy and fragmented. And one way of system biology to deal with that is exploiting biological networks, coming from public databases that annotates how molecules interact. And this information really provides the glue between the entities collected in multi-omics data, and moreover addresses the gap limits to the detection in multi-omics data. So in our research, we use public repositories that annotate interaction with the activity flow representation. So they annotate whether a regulator activates or inhibits a target. And the first type of resources, such as Omnipath or Signor, store this information in big interactomes, while other types of resources such as Wikipathways or KEGG will store the information in pathway maps. These two types of resources will prompt a different type of computational methods. So the first category of computational methods that is primed by interactome-based databases will return a big network with high coverage, but low interpretability due to the size of the interactomes. While the computational tools that are primed by pathway databases will return networks that are fragmented, where each fragment comes from a different pathway, but they are more interpretable because pathways usually ends on cellular functional traits. So our goal was developing a pipeline taking the best of both worlds. So returning a big network with high coverage, but also high interpretability. Moreover, we wanted the entities in the networks to have an activity derived from multi-omics data to make these networks context-specific. So we developed the SignalingProfiler, which is an R package that takes as input multi-omics data, so genomics, bulk or pseudo-bulk transcriptomics, proteomics, and phosphoproteomics collected between two conditions of interest, like disease versus healthy or treated versus control. And these experimental data are combined with the prior knowledge about how molecules interact stored in Signor and phosphosites databases. The output will be a static picture of the signaling remodeling in your experimental data. So the starting point will be a set of perturbed proteins, and the endpoint will be phenotypes, so functional traits. And as you can see, the entities in the model have an activation status. And the edges connecting the entities have a signs representing the interaction that is responsible for that activity change. So the activity of the proteins in the model is derived in the first step of the pipeline from multi-omics data. So we infer the activity of transcription factor, kinesis, and phosphatases using footprint-based approaches, which considers the modulation on their target transcripts or phosphosites as a proxy of the activity. So what I mean is that if we focus on the NFK beta targets and we look at them in transcriptomics, we find them downregulated, we can conclude that NFK beta is not transcribing, so is inactive. And the same reasoning is performed for kinases and phosphatases looking at their target phosphosites. But to fully exploit the phosphoproteomics, we also developed the phosphoscore algorithm that considers whether a phosphosite is activating or inhibiting the protein it belongs. So basically, if we know that a phosphosite is activating the protein and it's found hyperphosphorylated in the data, we can conclude the protein is active. So these two scores are eventually combined in a final activity score. So at the end of the day, you will have a list of proteins with their activity, and we have information for both human and mouse. Importantly, we annotate the molecular function of proteins focusing on signaling. And this is very important for the second step of the pipeline where we aim at reconstructing signaling. So we developed this abstract idea of signaling where transcription factors are at the end of the cascade, kinases and phosphatases are in the middle, phosphorylated proteins in between, and optionally a user can select a set of starting proteins that are perturbed proteins that depends on the context of your experimental data. So if you're modeling treated versus control, it can be your drug targets, or if you're modeling a disease, it can be a set of mutated genes in your condition. So we want to connect all this protein using the information in Signor and Phosphosite Plus databases. But to restrict this big interaction to the neighborhood of the inferred proteins, we use a shortest path algorithm. So we have a naive network that is activity agnostic. What I mean is that two proteins will be connected considering distance and not causality. So it may happen that two proteins are connected with two paths with the same length, but different biological meaning. In this case, we have an activating and an inhibiting edge. But since both proteins are active, the right edge is the activating one. Okay, so to perform this choice automatically for the whole network, we exploit the Carnival algorithm, which will remove some edges, but also some proteins if no paths are available. So at the end of this step, we will have a protein-optimized network, but it's not so interpretable. So in the third step of the pipeline, we connect the protein network to a set of phenotypes available in the Signor database. To this aim, I developed the PhenoScore algorithm that will link phenotype to the protein and will also associate to each phenotype an activity score based on the contribution of different signaling axes on the phenotype. So this phenotype is inactive because all these axes are inhibiting it. And this provides a functional interpretation of the model, but also a kind of validation. In fact, if you are modeling tumor, you may expect the signaling axis in your model to activate proliferation and inhibit apoptosis. So once we developed the signaling profiler, of course, we evaluated its performance on different data sets. In particular, we selected these three data sets where the multi-omic profile of cells was characterized upon a different perturbation. For each data set, we manually curated a gold standard, so a set of proteins and phenotypes with their expected activity that should be in the model. And we evaluated the ability of signaling profiler networks to grasp this gold standard, measuring the precision and recall. And as you can see, we reached 0.8 in all the datasets. So this means that the results are biologically relevant. So in these slides, I just reported the signaling profiler exemplary results obtained from its application on a phosphoproteomics dataset obtained from HeLa cell lines treated and stimulated with EGF. So as you can see, with signaling profiler, we could infer more than 70 proteins from the sole phosphoproteomics. We can also infer the activity of transcription factors without transcriptomics because, as I was saying earlier, we consider the regulatory power of phosphosites on transcription factors. And we connected all these proteins in a network that represented the EGF-induced signaling remodeling. And as expected, importantly, we noticed that the signaling axis activated the cell growth and proliferation while inhibiting apoptosis and cell death. So the results are biologically relevant. In our opinion, the strength of Signaling Profiler is the interpretability of the model. In fact, the user can select one or more phenotype and inspect the functional circuits converging on those phenotypes. And these circuits have a phosphorylation resolution level. In fact, we map phosphoproteomics data on the edges of the model. And you can expect these networks to look for crosstalk between different pathways. Here you can see the crosstalk between mTOR pathway and MAP kinase pathways toward the activation of proliferation. And this inspection can really prompt some mechanistic hypotheses that can be validated in a low throughput manner. We did this a lot in our lab and in our research in very different contexts, in cancer, in particular in leukemia, but also on neurodegenerative diseases or metabolic diseases. But today I'd like to focus on a very recent publication. In particular, we applied Signaling Profiler in the context of chronic myeloid leukemia, and we used the Signaling Profiler network to support drug repurposing. So as you probably know, chronic myeloid leukemia is driven by the BCR-ABL fusion oncogene. So many targeted therapies have been developed. The first one was imatinib, but unfortunately, 20-30% of patients are still resistant to this targeted therapy. So to study this resistance, we established in our lab a resistant cell line. And to understand what was happening in resistant cells with respect to sensitive one, we did phosphoproteomics. And we compared these two conditions. We fed Signaling Profiler with this data and to reconstruct the pro-survival pathways in our resistant cells. And as you can notice, the output network is very big. So we needed a systematic way of prioritizing crucial protein for resistant cells. So I developed this scoring system that is called the druggability score to identify crucial proteins based on 3 criteria. So the first one is that a druggable protein should be on resistant cells. Second, it should have a strong topological impact on cancer hallmarks. So it should have a high number of paths inhibiting apoptosis and activating proliferation. And importantly, it should be central in the model. So this is the result of the druggability score, and proteins with a positive druggability score will be crucial for resistant cells. But to enhance the translational power of this analysis, we just focused on proteins that were targets of FDA-approved drugs for hematological malignancies that are highlighted in red. All the crucial proteins with an FDA-approved drugs were validated by my colleague and friend, Dr. Valeria Bica. Cell viability of sensitive and resistant cells was evaluated upon their inhibition. And as you can see, all of them induced cell death in resistant cells. And importantly, we also validated the opposite. So negative druggability score induced the opposite response, so stronger cell death in sensitive cells. So these results suggest that the topology of Signaling Profiler networks can be used to identify drug targets. But to make this long story short that was published just last week, we noticed that the strongest experimental effect was displayed by the FLT3 inhibition. So in our paper, we suggest that FLT3 inhibitors that are usually used in acute myeloid leukemia could be used in chronic myeloid leukemia. And we say this because we confirm this result also on patients' derived samples. So to conclude, today I talked to you about Signaling Profiler, which is an R package to infer the activity of signaling proteins and phenotypes and connect them all together in a context-specific signaling network that is interpretable. You can use this network to interpret multi-omics data and prompt mechanistic hypotheses. Importantly, I showed you an application focusing on using the topology of the model for prioritizing drug targets, and we are now moving to use the topology of the model for prognostic biomarkers identification. In fact, the PhD project of my colleague Veronica Lombardi is a patient profiler, so a scaled version of Signaling Profiler end-to-end patients' data and handle cohort network. So basically, we want to derive for each patient a network, and from the cohort pool of networks derive topologically driven prognostic signatures. So I'm done. I'm in time, so I'm happy about this. And I would like to thank both my supervisor, Francesca Sacco, with the wet lab, and Livia Perfetto for the dry side of our research. And of course, all of you for the kind attention, and I'm happy to take any questions.